are going to study the prawn appendages. To do this practical, we'll need a prawn which we have soaked in water in a basin. Then we have the dissection board on which we have already written the different appendages, so that when we are taking the appendages from the prawn, we will be pinning just opposite this. Then of course we need pins so that we can pin the appendages onto the board. We need a hammer with which we will hammer in the pins here. We need scissors, which is optional. What we actually need is a blunt forceps and also a sharp forceps. Sometimes you may need a needle, and it's optional to have gloves. Thank you. Ready? So first of all, you can see this entire ventral part of the prawn here. This part is the abdomen, and so it has got 1, 2, 3, 4, 5, 6 abdominal segments. So we have got the segments here. For each segment we have got a pair of appendages. Then the final segment, for which you will be having the uropod, which is attached to the telson. Then we have got the cephalothorax region. In the cephalothorax region we have got so many thoracic appendages. Okay, thoracic appendages. These are all the thoracic appendages. And then as we move towards the cephalic, the head region, we have got cephalic appendages. So when we do the cephalic, when we start doing, we have got two options. Either we can start from the beginning here, or we can start from somewhere here. So what I am going to do is, I am going to start here at the second maxillipede. I am going to start here at the second maxillipede. I am going to start here at the second maxillipede. I am going to start here at the second maxillipede, because the second maxillipede, it has got a very notable question mark sign. It has got a very notable question mark sign. The second maxillipede, it has a very notable question mark sign. So for studying the appendages, so this is the abdominal part, the thoracic part. So in the abdominal part you have got 1, 2, 3, 4, 5 segments here. The sixth segment, which has the uropod and the telson here. Then these are all the swimming legs. Then in the thoracic region we have got the walking legs. So after the walking legs we have got the cephalic appendages. So the first step here is to find out the second maxillipede. So I am very carefully moving all these appendages back. I am not breaking anything here, just allowing my hand to hold them back, and you can see the second maxillipede here. So what is the speciality of the second maxillipede? The speciality of the second maxillipede is the question mark. So if you look like this, you can see this as a question mark. See, the endopodite has been in the shape of a question mark. The endopodite is in the shape of a question mark. So with this we know that this is the second maxillipede, because of the question mark like endopodite. So we know this is the second maxillipede. So from here onwards it is easier to identify when we go to the cephalic appendages, because if you are trying to break this, it is very small, and if we try to take one out, it may cause so many mistakes. So better to start from here, that is the second maxillipede. So I have identified the second maxillipede here. I am just holding it back on this side. Now let us see the next one, that is the first maxillipede. So I just lift it out very carefully. I am lifting it out. You can see the different parts, and I am using my heavy forceps to remove it. So I am going deep like this, to pull it out, going deep like this to pull it out. So I get the first maxillipede here, the first maxillipede here. Okay, then after the first maxillipede, the second one is the elephant ear like. So it's shaped like a elephant ear. So that is the second maxilla. That is the second maxilla. You can see here the different parts of the second maxilla. We have to be extra careful not to break anything out. So I'm just lifting it up like this, and again I am using the forceps to catch it at the base and pull it out. So yeah, second maxilla. So some part of it is slightly torn here, but there's nothing which we can do. Next you can see it is just almost leaf like, and that is the mandible here. So just pulling all of these back. Ready? So we have taken all the cephalic appendages, the minor ones, the small ones. Now what we have to do is we have to take the antennae and the antenules. So here you see this entire thing. This is the Endopodite. You can see this is the Endopodite, the Exopodite, and you can see how the filamentous process of the antennae is. So this entire thing has to be taken here together. So you literally can use your fingers to squeeze it, because it may be difficult for us to take this entire thing with the forceps. So see, the entire thing has to be taken out for the antennae, and then when you look here, we have the antenule. We have the antenule. So in the case of the antenule also, you can see there is an entire process here, So we have to take all these things together. We have to take all these things together. We have to make sure that all these are present for the dissection in the display. So all the cephalic appendages are done, and the first maxillipede, all the cephalic appendages are done, and the first maxillipede is also done. Now let us go to the second maxillipede. Remember the question mark shape. So it is still waiting for us here. So when we go back here, we can see the question mark here. It is waiting for us. The question mark, it is waiting for us. So we have to take the entire filament here and see, there is a small epipodite on it. There is a small epipodite on it. So when we take this, we have to take the entire thing from the base. If you don't take it that way, something will be lost. So I'm keeping it here. Then we come to the next one. So here you can see the epipodite here. See, you can see the epipodite here, and you can see that these two parts, the endopolite and the exopodite, along with the basa and the coxa, all these things have to come in the display. All these things have to come in the display. So you pull it out from the root like this, and we have kept it here. Then we have got the legs. So if you are looking here at the end, you can see how the tip is it not? It is like a scissors. So that these are the chelate legs. So the chelate legs also have the epipodite. They also have the epipodite. So be very sure that all this is maintained here when you are pulling it out. The first chelate leg. After the first chelate leg, see the epipodite here, the second chelate leg. See the end of it here. See the end of it here. So I am pulling it out like this. Then we come to the final chelate leg here. Here also it has the epipodite. So just see here, the epipodite is here, and make sure the epipodite is coming with it. Then we come to the non-chelate legs. We come to so here we have the final two walking legs, the non-chelate leg. So that also we'll just pull in from here. The second one and the first one. So now when we look in here, we have got the swimming legs. So one thing which we have to remember here is, if the animal here is a male, then this then this will be fused together like this. So now you can see it is not fused together, it is separate like this. So it means that it is a female. It is a female. So again I am removing the leg only from one side. I am removing the leg only from one side like this. This is the first abdominal leg. Then similarly we have got the second abdominal leg. So from this place onwards things are quite simple because they are all very similar. So anyway I'll just take them out here. The third abdominal leg, the fourth abdominal leg, and the fifth abdominal leg. So here I have taken only one side. This I have left intact. Okay, so here you see I have taken only one side because the rest of them are here. Okay, this side is here, this side is here, only this part is on the animal. Finally we have got the Uropodia. This we have to take. Taken like this and kept like this. So we have already seen all the prawn appendages, the 5 cephalic, the 8 thoracic and the 6 abdominal. Now right here when you look at the antenule, you see this part here, this is the precoxa. Then you have the coxa and the basis and the 2 flagellar processes. This part is known as the protopodite. Then when we come to the antennal part, this is the filamentar region. If we focus on this region will find the basal segments here. Then down here we have got the basis and the coxa, and this is the exopodite. Here. So this is actually supposed to be the derivation of endopolite. Next we come to the mandible. When we come to the mandible, you can see a small hairy something, a leaf-like structure which is protruding here. This leaf-like structure is the palp, and then the palp is connected to a calcified base, and if you hit your needle here, you can actually feel the teeth. There is something quite hard here. So you can actually feel the teeth here. Next we come to the first maxilla. When we look at the first maxilla, we have got a feathery endopodite, and then we have got the basis and the coxa, which together consist of the protopodite. Next we come to the second maxilla. The second maxilla is an elephant-eared structure. It is an elephant-eared structure. You can see this very large elephant-eared structure, which is actually the exopodite. This elephant-eared structure which I am lifting here, this is the exopodite. And when you are looking in here, you can see so many different things here. This first one, this very small one here, this one is the endopodite. And then after this endopodite, you can see a bilobed structure here. You can see this bilobed structure here, and that together consists of the protopodite. Next we come to the first maxillipede. When we come to the first maxillipede, here you can see this exopodite here. You can see how this hair-like structures are here, and then you have got this filamentous endopodite. Then you have got the protopodite here. You can see the protopodite and the epipodite. This is the epipodite. So this is the first maxillipede. Then we come to the second maxillipede. This was the appendage which helped us to take all the cephalic appendages. This is the marker here. So when you look at it carefully, you can see this question mark symbol. Okay, now I think it is very clear. This question mark symbol, this is actually the endopodite. The exopodite is one straight structure. This is the endopodite here. Then you have got the epipodite. This is the epipodite. And for your examination purpose, if you don't get the epipodite, you will not get full marks. So this is the epipodite here. And all of this is fixed onto the basis and the coxa. Next is the third maxillipede. And here also you can see the epipodite, the endopodite and the exopodite, and the basis and the coxa, which actually is fused together to form the protopodite. Next we come to the walking legs. So remember here, from here onwards, okay, so from here onwards we are talking about the thoracic appendages. So it was till the antenule, the antennae, the mandible, the first maxilla, the second maxilla, which consisted of the cephalic appendages. From the first maxillipede onwards, we are talking about the thoracic appendage. Then we talked about the question mark symbol, which is present in the second maxillipede. So when you are doing the dissection, if you can identify this and you keep this back as an identifying marker, from here onwards it's easy to pluck each one out and keep it in the correct position. And when for the exam purpose, whatever is being asked, you only need to pin it and display in the finally. The rest you can remove from the display. So then we come to the third maxillipede, and we also have the walking legs. So when we are talking about the walking legs, there are two types, the chelate and the non-chelate. So when you are looking at the chelate and the non-chelate, what we actually find is that in the case of the chelate, the distal end, the segment in the distal end, it is claw shaped. You can see a scissor like there are two parts here, two components here, and these two components is known as the dactylus and the chela. So this is actually divided here, but when you are looking into the non-chelate legs, we do not have any such claw shaped structure. So when we talk about the chelate legs, the walking legs, again you have got the epipodite. You can see the very light, almost transparent epipodite here. Then you have got the protopodite. Then you have got the different segments, namely the ischium, the meris, the carpus, the propodes and the dactylus. And it is the propodes and the dactylus which forms the chela. It is same here for the others also, for all the other chelate legs. The second and the third chelate leg also has an epipodite, and then they all have these different parts, namely the ischium, the meris, the carpus, and finally when you come to the last part, that is the chela part, you can see how it is being split here. See, so that is the propodes and the dactylus forming the chela here. And in the case of the chelate leg, non-chelate leg, we do not have anything like that. We do not have an epipodite also. And we have a very small exopodite. Which is reduced in nature. When we come to the abdominal segment, for abdominal appendages, for every segment we have got one pair of appendage. The first pair is very important. Because in the case of the males, we see that the endopodite is fused. On either sides the endopodite is fused. So in the case of the male, the abdominal region, there are 6 appendages, 1 pair each for each of the segments. So the first pair is very important because it helps in sex determination. In the case of the female, we have got a basal part, the protopodite. So that consists of the coxa and the basis, and there is also an exopodite. There is a very reduced endopodite here. A part of the endopodite here is broken actually, so we cannot see the entire thing, but you can see a very small feather like thing here. You can see the small feather like thing here. It's slightly bigger. It was broken in the specimen here. In the case of the male, that small projection, the endopodite, it is somewhat larger and it fuses together. So these are the first abdominal appendages of the legs. So it fuses together like this to form a structure which is known as the petasma. So if you see a prawn having this in the first abdominal appendage, that means it is a male. But in this case it does not have it, so it is a female. The second to the fifth one, they are all same. So second, third, fourth, fifth, they are all same in appendage. All of them have got the base, that is the coxa, the basis, then the exopodite and the endopodite. The exopodite and the endopodite. So that is same for all of the other abdominal appendages. And finally for the sixth segment, you have got the uropod. The uropod has an exopodite and an endopodite, the exopodite and the endopodite, and they are connected together here in the middle by a sympod. So this is all about abdominal appendages. So just refer to your textbook and you will be able to study more about it. So that is all. Thank you.